So today's topic of discussion is actually some non-receptor tyrosine kinases which are being in association with these tyrosine kinases. So this we have discussed very uh, briefly. In today's class, we will go into the details how these complexes are regulated by JAK-STAT pathway of cell signaling. So earlier in previous class, actually, I told you about these JAK or the genus kinases, but only the introduction was given in that particular class. So before starting even the genus kinases, actually, I will remind you to the class in which we have started tyrosine kinases actually. So receptor tyrosine kinases were three major classes. One class is a single transmembrane protein, for example, epidermal growth factor receptor. The second class is insulin receptors, for example, which are having alpha and beta subunits, where alpha is for ligand binding and the beta subunit is having this catalytic domain, okay, in the cytosol. And the third class is platelet-derived growth factor receptors, which do not having these cysteine-rich domains, extracellular domains, whereas they are having some immunoglobulin-like domains or antibody-like domains. So these are three type of receptors. These are called protein tyrosine kinases or PTKs actually. So this we have discussed. And the second thing is also very important about these receptors. They are having about 100 amino acid stretch, which is interfering with this particular catalytic domain. So this catalytic domain, this is called receptor tyrosine kinase. This domain is having some kinase function. So this is not continuous. This is having some interrupted amino acid sequence with unknown function. This is not having some catalytic activity actually, but it is present in these type of receptors. So based on structure and certain properties of these receptors, they can be classified into three major families. Okay, actually these are the same family, receptor tyrosine kinases, but these are subfamilies. Now, for example, epidermal growth factor receptor, which is a single polypeptide, will form a dimer after formation of growth factor or epidermal growth factor. This dimer formation will undergoing some autophosphorylation or cross-phosphorylation event, so that this catalytic domain can phosphorylate some of the residues. Which residues? Actually tyrosine residues specifically in the extra catalytic domain, so that certain protein can come and bind to these phosphotyrosine residues. Now the question is, which proteins will bind to this phosphotyrosine residue? So obviously this has been repeated several times, and only those proteins which are having some SH2 domains will come and bind to these phosphotyrosine residues, as shown in the next diagram. So this is a receptor tyrosine kinase, and it is forming a dimer after binding with the growth factor, and it is showing a fully functional catalytic domain, which is phosphorylated over tyrosine residues, not only in the catalytic domain part, whereas it is also phosphorylated extra catalytic sequence of this particular receptor protein. So these phosphotyrosine, which are present outside these catalytic domain, outside this red colored section, so they are having some binding role. So they will create some docking sites for the proteins which can come and bind to these phosphotyrosine specifically. And these proteins are some downstream signaling molecules which will transfer your signal from this receptor inside your cytosol of the cell, and they will have these characteristic SH2 domains, which are called SRC homology 2 domains actually. Okay. Now, there is two type of classification from this point of view. One type of receptors or receptor tyrosine kinases may have a catalytic domain. अब देखो दो तरह से इन्हें क्लासिफाई कर सकते हैं. एक रिसेप्टर्स ऐसे होंगे जिनमें ये रेड कलर की सीक्वेंस होगी, यानी कि जो काइनेज एक्टिविटी को शो करेंगे, और दूसरे होंगे जिनमें ये कैटालिटिक डोमेन एब्सेंट होंगे, मींस दे डू नॉट हैव दीज रेड कलर स्ट्रेचेस, मींस दे they do not have a functional receptor tyrosine kinase domain. Okay, so now the question is, if they do not have these receptor tyrosine kinase domains or red color stretches, then how they will be able to transmit the signals inside the cell? So to transfer the signal and to do some receptor tyrosine kinase activity, to perform that particular activity of these red colored stretches, some other proteins which are not the part of the receptor, so they're called non-receptor tyrosine kinases. These are certain proteins, and one example of these proteins is actually the Janus kinase, JAK, which we are going to study in this particular class. वैसा ही एक प्रोटीन है जिसको बोलते हैं Janus kinase, and this Janus kinase is given in this particular text. So if the catalytic domain is absent in the receptor, so certain protein which are called non-receptor tyrosine kinases will form some non-covalent interactions with these C-terminal domain of these receptor tyrosine kinases, and they will perform the catalytic function inside the cytoplasm. So they will phosphorylate the tyrosine residues because they are also tyrosine kinases. So their targets are tyrosine amino acids either in the proteins or in these non-receptor tyrosine kinases or Janus kinases. For example, it is given here the kinases associated with the cytokine receptors belong to Janus kinase, and we will study in detail how these Janus kinase family will interact with another transcription factors called STATs. That are the STATs are actually the signal transducer and activators of transcription. So you can find some, okay, so it is given here. So these tyrosine kinase associated receptors are actually these cytokine receptors, and the family of non-receptor tyrosine kinases are called JAKs or Janus kinases. These Janus kinases will activate and phosphorylate some of the regulatory proteins, which are also, these are gene regulatory proteins. So these are generally transcription factors. So these are called STATs for signal transduction or transducers and activators of the transcription of the mRNA formation. So these STAT proteins will translocate into the cytosol. These are actually present in the cytosol, but they will migrate into the nucleus. They will be translocated inside the nucleus, and after migration, they will regulate certain genes transcription even. Okay, so these transcription of certain genes will be under the regulation of these okay, STAT proteins, which are the signal transducer and activators of transcription. Now, I hope this is clear in your mind what we are going to study in this particular class. So we are starting our discussion of receptor tyrosine kinases. So if you are not still able to get the introductory portion of this particular lecture, I will explain again. So if you are having if you are facing any queries, you can write in the chat box. Okay, so we are starting with Janus kinases and their regulators or the transcription factors, which are called STAT proteins. So JAK and STAT proteins will overall control somehow your signaling cascade and certain genes which are under the regulation of STAT proteins, because STAT proteins are transcription factors and they will regulate the transcription overall. Okay, so these cytokine receptors, for example, are responsible for signaling in certain immune cells. For example, interleukin-2 is responsible for T cell proliferation. Erythropoietin hormone, as you have studied in your bone marrow, it is required for RBC production, and certain other hormones which are also the polypeptide growth hormone, for example. Okay, so they are doing signaling via the receptor tyrosine kinases, and after the receptor tyrosine kinase in the cell cytosol, these Janus kinases, for example, which are also called non-receptor tyrosine kinases, will transmit your signal to the STAT proteins. And okay, so we are moving to some other page where this pathway is given actually. So this we have discussed. So I'm scrolling very without discussing all of these. Today we will discuss this actually. So this is called JAK-STAT pathway. So here you can see one cytokine is already bound to your dimer, And this dimer is, these receptors are called receptor tyrosine kinases, but you can see here they are not having any red color portion or domains here. So therefore they do not have any intrinsic these are called tyrosine kinase activity. Therefore they will associate with some of the JAK proteins. These JAK proteins are called Janus kinases proteins. So now the question is very important question, how these protein will interact with these receptors. So actually the type of interaction between the two proteins, either one is this receptor and second is these Janus kinase proteins. So this kind of interaction is generally non-covalent interaction. So generally these non-covalent interactions are also found in case of your antigen antibody interactions, for example. Similarly, these two protein will interact, and these kind of interactions are non-covalent primarily. Okay. So once the cytokine will bind to the extracellular surface or N-terminal domain of the receptor tyrosine kinase, there is a conformational change in the C-terminal domain, which will lead to the activation of these Janus kinases. Once these Janus kinases will be activated, they will phosphorylate the receptor outside the section where they are actually interacting with the receptor. So these phosphate residues from the ATP actually, the gamma phosphate will be transferred to these receptor proteins. This is section of the part of these receptors only. So these will create the binding site for these proteins. These proteins are called STATs, signal transducer and activators of transcription. So the two STAT molecules will be phosphorylated by these Janus kinases. Okay, so Janus kinases are doing two type of activities here. First, it is phosphorylating the tyrosine residues in your receptor itself. Second, it is phosphorylating your STAT proteins. Okay, so these STAT proteins also having some SH2 domains. So with the help of these SH2 domains, these can interact directly with these receptors, so that they will come in the close proximity of the Janus kinases. And once they were in the close proximity, they are able to phosphorylate the specific tyrosine residues over the STAT molecules, so that the STAT molecules can be fully functionalized. And these type of phosphorylations will lead to some conformational changes in these STAT molecules, so that they can create the binding site for each other, so that these phosphotyrosines over these STAT molecules and these SH2 domains will specifically bind with each other, and this kind of specificity will lead to the formation of dimers. Then this dimer will be translocated inside your nucleus, and in nucleus they will bind to certain response elements over DNA, so lead to the transcription of certain target genes which are under direct regulation of these STAT transcription factors of proteins. So this is very simple to understand, and hope this is very, very clear to you all how this kind of signaling is being happening. Okay. So they are non-covalently associated with your receptor tyrosine kinases. Like receptor tyrosine kinases, the tyrosine kinase associated receptors contain an N-terminal extracellular ligand binding domain, as we have seen. So they will bind with certain cytokines, single transmembrane alpha helix. So always whenever there is a transmembrane section is always alpha helix in nature, and they will have some certain hydrophobic residues so that they can interact with the lipids in the your plasma membrane. And the C-terminal cytosolic domain, so this is not having certain catalytic activity. So this is the cytosolic domain which is interacting with your Janus kinase. However, the cytosolic domain of the tyrosine kinase associated receptors are devoid of non-catalytic activity. So this is also important to note here. Sorry. Okay, so these will not have any non-catalytic activity. Instead, these receptor function in association with non-receptor cytosolic tyrosine kinases, which are called JAK kinases actually. so which are activated as a result of ligand binding. So this ligand binding will cause some kind of conformational changes and will help in the activation of these genus kinases, which will interact non-covalently with your receptor tyrosine kinases. This is very clear. A variety of receptor classes belong to this particular category, these are same kind of for example, receptor for antigens. Receptor for antigens are present over antibodies and interleukins on lymphocytes. So they are also doing signaling in the same way. Also, the integrins, we will study in today's class how these integrins, which are present. So these integrins are having role. So your cell in the body are interacting with extracellular matrix with the help of the two family of integrins, either alpha and beta integrins. So I will show you how these integrins are present. Actually, it will come in the text somewhere. And receptors for various cytokines and some polypeptide growth hormone, for example. Okay, so they will do the signaling via these JAK-STAT pathway, which we will discuss in today's class. The largest and most diverse class of receptor, these tyrosine kinases associated receptor, is called cytokine receptors. So you need to remember this also. These cytokine receptors are forming the largest class of non-receptor tyrosine kinases. Okay, so you will remember all these things. Now the interleukin 2, erythropoietin hormone, growth hormone, and one another is prolactin hormone. So all these are doing signaling via these JAK-STAT mediated signaling pathway, whose cytosolic domains are closely associated with the members of the family of cytosolic tyrosine kinases. These tyrosine kinases are present in the cytosol, but they will be recruited. They will bind non-covalently to the receptor molecules, so they will be recruited to the plasma membrane. Okay, so the JAKs phosphorylate and activate the gene regulatory proteins, and these are called STATs, which are the signal transducers and activators of the transcription. These STAT proteins are located in the cytosol, and after activation, they will migrate into the nucleus. They will migrate into the nucleus as a dimers, and then they will regulate the gene transcription of certain target genes. Now, how this JAK-STAT pathway is mediated. So the JAK kinases take their name, actually they're called genus kinases, from the characteristic presence of the two kinase domains in each molecule. So you need to remember there is two kinase domain in each protein, so that they will able to phosphorylate both your receptor as well as the STAT protein. So they will have two kinase domains in each JAK molecule. Several members of the family are known, including the three JAK, 1, 2, 3, and one another related molecule that is called TYK2, which is also coming under the same family of JAK proteins. So each associated with a specific set of cytokine receptors. Now, these are different kind of tyrosine kinases, so they are having their specific set of cytokine receptors. The JAK kinases are tyrosine kinases whose major substrate are transcription factors, and you need to remember these are transcription factors, which are also the gene regulatory protein, which are called STAT proteins. Actually, these are several types of STAT molecules, and each STAT protein is phosphorylated by a particular set of JAK kinase. So we know there are certain kind of JAK proteins, and they will interact with certain type of cytokine receptors, which are receptor tyrosine kinases or non-receptor tyrosine kinases, for example. And they will activate specific class of STAT molecules, all STAT proteins containing. And now it is very important again to note this, because these SS2 domains are required so that the phosphotyrosine in the which is present in the receptor or the present in the STAT itself can interact with each other. In the receptor, cytosolic domain is a central DNA binding domain, okay, and a C-terminal domain. So it is actually telling about STAT protein. So the STAT protein is made up of N-terminal SS2 domain. It is having some central DNA binding domain because it is a transcription factor and will go inside the nucleus controlling certain target genes. So it will have a central DNA binding domain with which it can interact with your DNA, and C-terminal domain with a critical tyrosine residue. So the tyrosine residue is present over your C-terminal domain. Okay, so where it will be phosphorylated. Now, So the ticks where I have added these green color ticks are the possible questions you should remember and you need to underline in your respective books. Okay, so now we are moving to next page. Okay, so this is very quiet and similar explanation in your Pathfinder as we have studied from the Cooper. So once the stat is bound to the receptor, the C-terminal tyrosine is phosphorylated because this specific tyrosine is present over the C-terminal and it will be phosphorylated and associated JAK kinase by an associated JAK kinase. So see here, this is your receptor tyrosine kinase, which is a monomer. It will form dimer after binding with certain cytokines, for example, interleukin 2, erythropoietin hormone, growth hormone, or prolactin. After dimerization, they will interact with certain JAK kinases, which are present in the cytosol. These JAK kinases will be interacting with these receptors. These are called receptor tyrosine kinases. The second major class of cell surface receptors, and these are present in the cell cytosol, so they will start phosphorylating each other, and also they will phosphorylate your tyrosine residues which are present in your receptor itself. Once they will phosphorylate the tyrosine residues, so they will creating the binding site for these proteins which are called STAT proteins. These STAT proteins will have certain certain okay certain where is my uh, text? Okay, so these STAT proteins will have certain domains which are called SH2 domains. So these SH2 domains will specifically interact with the phosphotyrosine residues, and then these uh, these JAK molecules or JAK kinases also phosphorylate the STAT proteins so that they can interact with each other. So these phosphorylated forms also form a dimer and they will enter into the nucleus. Okay, so after going into the nucleus, they will regulate certain genes. Fine. Sorry. So actually, before starting another class of serine threonine kinase receptor, that is your tyrosine. Oh, sorry. This is TGF beta, transforming growth factor beta. Let me finish this portion from your Cooper also, so that we can note certain things which are present. Okay. So somewhere. So we have discussed PI3 kinase, and this PI3 kinase is actually activating your protein kinase B or AKT as we have discussed. MAP kinase pathway, we have yesterday focused over MAP kinase pathway. So MAP kinase is having certain proteins, RAS, RAF, ERK, and these proteins. Okay, so ERK is your MAP kinase actually. Okay, so they will activate each other, and this pathway are examples of indirect connection between the cell surface and the nucleus, where they will transmit signal via certain different class of molecules. So see here, this growth factor is activating RAS protein. RAS protein is certain G protein, actually GTP binding protein. Then it will activate the RAF, MEC, ERK. ERK is actually the MAP kinase, and this MAP kinase will go inside the nucleus and will control certain genes. So see here, this is the same receptor tyrosine kinase, but actually this is the real receptor tyrosine kinase because it is having certain catalytic active sites. So this is the receptor, and these are tyrosine kinases domain in this receptor, so they can function actively. But because JAK-STAT pathway is a kind of pathway where these receptor tyrosine are not having the kinase domains in the cytosolic portion, so they will interact with some other protein called JAK proteins or Janus kinases, which help in activation of phosphorylation of tyrosine residues. So in this pathway, this is called MAP kinase pathway because this ERK is actually called MAP kinase, and this MAC, which was a dual specificity kinase, which is either a threonine kinase or a tyrosine kinase, is a kind of kinase which will phosphorylate the MAP kinase. So this is MAP kinase, and RAF protein is one more kinase. So this is MAP kinase. So Okay, so there are 3K, there is 2K, and there is only 1K. This is kinase actually. This kinase will go inside the nucleus and it will phosphorylate because it is a kinase. So, and it is a serine-threonine kinase, so it will go inside the nucleus. And certain cytosolic proteins also it will able to phosphorylate. So this kind of signaling is a kind of indirect signaling where this particular receptor is with the help of certain different kinase proteins is actually able to transfer the signal inside the nucleus. But JAK-STAT pathway is a kind of pathway where the proteins are directly recruited and they will go inside the nucleus. So, for example, there is only a single step. So, this receptor, these are non-receptor tyrosine kinases. Okay, sorry. These are non-receptor tyrosine kinases because they are not having these red color catalytic domains. Okay, so they will directly phosphorylate and activate STAT proteins, and there is no intermediary proteins. These STAT protein will translocate directly inside your nucleus and will be activated. Okay, fine. So these are more direct connections between the growth factor receptor and the transcription factor, because directly the transcription factor will be activated and going inside the nucleus, in which the targeted transcription factors or STAT proteins are phosphorylated and directly going into the nucleus directly by the receptor associated protein kinases. These are called JAK kinases or Janus kinases. Now, the key elements in this JAK-STAT pathway are called STAT proteins. As we have discussed in the cell cycle, the two type of proteins which are called cyclin CDK complexes, where the cyclins are regulatory proteins and CDK are kind of CDK are some catalytic proteins. They are doing some function. These regulatory proteins in this particular pathway, JAK-STAT pathway, are called STAT proteins. So, signal transducer and activator of transcription, which were originally identified in the studies of cytokine receptor signaling, as we are seeing in this particular diagram. The STAT proteins are a family of 7 transcription factors. So, so there are certain. Okay, let me. So they are having 7 members in this STAT family. I don't know what is. So, okay, so they are having 7 members. Now, okay, so 7 transcription factors. That are containing the specific domains, SS2 domains, so that they can interact with your phosphotyrosine residues. They are inactive in unstimulated cells where they are localized to the cytoplasm, but they will translocated into the nucleus after being activated by certain cytokines or some growth factor binding. Okay, so stimulation of cytokines receptor lead to the recruitment of STAT proteins, and they will bind by their SS2 domains to the phosphotyrosine containing sequences in the cytoplasmic domains of the receptor polypeptides. Okay. Following their association with the activated receptors, and the STAT proteins are phosphorylated by members of JAK family kinases. So everything which we have already discussed and given this particular text, you can go through and you will find that actually everything which is written in text we have discussed. Okay. Now the second family is second kind of signaling, which is the topic of this particular class, is transforming growth factor beta signaling. And this particular signaling is happening via the two kind of receptors. One kind of receptor is called one, and second kind of receptor is having this. So they are quite different. So in this pathway, actually, this transforming growth factor beta is able to interact with these two receptors. They are forming heterodimer, and in heterodimer, the second receptor will phosphorylate the first receptor, okay, at a specific residue. And after phosphorylation, there is certain conformational changes in this particular receptor so that it is able to phosphorylate the SMAD protein. So this particular protein is under the control, or it is a target protein of this one receptor. So two is activating one and one is activating the SMAD protein. So it is actually phosphorylating. After phosphorylation, it will again undergoing some conformational changes and it will be activated. So this SMAD protein will be translocated inside the nucleus and will able to activate the transcription of certain targeted genes directly. So this kind of signaling is also a direct kind of signaling where there is no certain no intermediary protein kinases. It is directly activating the SMAD protein, and SMAD is acting like a transcription factor. The transcription factor will be activated directly by binding of these growth factors, and thus the gene regulation or gene transcription will be happening. So the receptor for members of transforming growth factor beta family of growth factors are the protein. These are called serine threonine kinases. So you need to remember these things, which are very important to remember. If these are serine threonine kinases or tyrosine threonine kinases or something else. Okay, so mostly they are serine threonine kinases, but still you need to remember whether they are okay. So this transforming beta growth family factor, these kinases are actually serine threonine kinases. So obviously they are serine threonine kinases. So this particular residue is either serine or threonine. Similarly, this particular residue which is phosphorylated in SMAD is either a serine or threonine. So they are directly phosphorylating these transcription factors, and these called SMAD family. Okay, so the receptors are composed of two type of polypeptides. One is called type 1 and type 2. They were forming a heterodimer. Type 2 receptor then phosphorylate the type 1, and the 1 will phosphorylate your transcription factor, which is in turn phosphorylate a SMAD protein. Now the SMAD protein, phosphorylated SMAD then translocated into the nucleus, and they will regulate the gene expression directly. So this is also a very simple kind of cell signaling directly. Your transcription factor is being phosphorylated and activated, which is controlling certain genes in the nucleus. It should be noted that there are 42 different type of members of transforming growth factor beta family in humans. If you note in humans, there are 42 different kind of transforming beta family in humans only. Okay, so in certain other may possible in other kind of organisms also, but in human there are 42. Now, one more thing you need to remember from the previous discussion, actually, this particular thing, actually, this we have discussed that certain hormones. Okay, so they are doing signaling via JAK-STAT pathway because this is growth hormone. Similarly, the prolactin hormone, interleukin 2, and erythropoietin hormone are mediated by JAK-STAT pathway of cell signaling. Okay, so this you should remember. This may be a direct question or okay. Now, responses in the target cells. This is accomplished by the combinatorial interaction of the 7 different type 1 receptors and 5 type B receptors. So actually type 2 is activating the type 1, and there is different type of diversity present in our cells because this transforming growth factor receptors are 42 types. So they can form different type of combinations because there is already 7 types of type A and 5 types of type 2 receptors. Okay, so which lead to the activation of different members of SMAD proteins, a total of 8 family members. Now these receptors can activate different families of SMAD proteins which are controlling different genes. So there are 8 families of these SMAD proteins already reported in your cells. SMAD family members can also be phosphorylated by ERK. Now this is very important to note here that something unusual is again happening. Okay, so actually I need to use this. So these SMAD family protein is somewhere interacting with another type of signaling that is called ERK or MAP kinase signaling. As you uh, remember from the previous discussion, these are called MAP kinase family. Okay, so MAP kinase signaling. Now you need to note here that this SMAD family, which is doing signaling in response to transforming beta growth factor, and this receptor tyrosine kinase can also do signaling via these MAP kinase pathway, in which the RAS, RAF, MAP, RAS, RAF, MAC, and ERK proteins are being activated. And this type of intersection between these two type of pathways, because these are two completely different pathways of cell signaling, now it is showing that some kind of intersection between these two type of cell signaling pathways. So one is transforming beta growth factor SMAD pathway, and second is your MAP kinase pathway. So it is playing some critical role in embryonic development. So this, if there is two type of signaling and they are intersecting each other, not only this, a certain other type of signaling pathway can intersect each other, and this is actually happening in our cells, because in cell there is a large diversity of these proteins and they are crossing each other. So as we will see in our next discussion of this cell signaling class. Okay, so now I'm raising this and I'm moving to your pathfinder so that we can discuss certain important points. Okay, so these are called serine threonine kinases. As we have discussed, there are two types. And the new thing which you should note from your pathfinder is there is transforming beta superfamily, and there is large number of structurally related secreted dimeric protein. As you have noted, there is 5 and 7 types of type 1 and type 2 receptors. This superfamily include transforming beta growth factor themselves, activin proteins, and the largest family is of bone morphogenetic proteins, that is called BMP, which constitute the largest family. Okay, so this BMP protein is actually the largest family in your cells. Each member of transforming beta superfamily binds to the characteristic combination of type 1 and 2, able to phosphorylate some protein, which are called SMAD proteins. Activation of SMAD causes their translocation from the cytosol into the nucleus, where they are controlling the gene expression. Okay. So we are not going into the details of other pathway, which is given in your pathfinder. Before starting nitric oxide signaling, we are having NFkB signaling, which is a kind of signaling happening inside your immune cells. Okay, so I'm underlining some of the important points in this particular text. Now, the third kind of signaling in today's class is called NF-kappa B signaling. What is this NF-kappa B is actually the transcription factor. This signaling is another example of signaling pathway that directly target the specific family of transcription factors. So these are transcription factors. These family consists of 5 transcription factors. Okay, so that play a key role in the immune system. So these type of signaling, as I already told you, is actually happening in the immune system cells, which we will study in details in our immunology class. Okay, and in inflammation, because this is happening in your immune cells, so it is taking part in inflammation as well as regulation of proliferation, survival of many kind of uh, immune cells, actually animal cells. Members of this transcription factor family are activated in response to variety of stimuli, example cytokines, growth factors. There is viral infection, so this is pure immunology here, and if there is a DNA damage in the cell, so this will lead to the activation of NF-kappa B. An unstimulated cell, actually, so I will show you what is happening inside your cell. So let me remove all this and scroll down so that, sorry. So see here, so this is your NF-kappa B signaling, where there is an inhibitory protein. So you can note here certain things directly so that you can better understand. Okay, so this is text and I'm writing it. Inhibitory protein. So actually this I-kappa B kinase is a kind of inhibitory protein which is interacting with your your NF-kappa B. So if a signal will come, for example, some viral infection is happening or some DNA damage is there, so signal will come and it will lead to the phosphorylation of your inhibitory protein that is called I-kappa B, which is inhibiting your NF-kappa B complex. Okay, so it will lead to this phosphorylation will lead to some conformational change, and then the ubiquitin molecules, ubiquitin protein. So this will undergo ubiquitin mediated proteasomal degradation. So ubiquitin will uh, form a polymer. This polyubiquitin chain will be added over this I-kappa B, so that this will be degraded specifically by a proteasome that is called 26 as proteasome. Sorry, this is 26 as proteasome. So it will degrade this particular I kappa B. So it will free NF kappa B. So these nuclear factors or the transcription factors will, as a dimer, will move inside your nucleus so that they will able to control certain gene transcription inside the nucleus. So see this, they will move through the nuclear pore complex, and inside the nucleus they are able to activate the transcription of certain genes which are under the control of this NF-kappa B. Okay. Now, beside this kind of signaling, there is another kind of three different signaling that are called Hedgehog, Vent, and Notch pathways of cell signaling. And this is happening inside your embryonic development. So whenever there is embryo in early embryonic stages, these cells will showing these type of signaling. Okay, so let me choose something. Okay, so these hedgehog and vent pathway are closely connected signaling systems. And the third is notch pathway, which is quite different. So these are very related, but they will have, all the three pathways are different because they are having different names, and they are having different regulatory proteins which are doing the regulation, but they are something connected, means something connected in the sense they are similar in nature, play a key role in determining the cell fate during embryonic development. So if there is early embryonic development, so they are all involved in these early embryonic developments in determining the cell fates during early embryonic development periods. So both Hedgehog and Wentz pathways were first described in Drosophila, but members of these families are also reported in mammals or in humans also. Okay, so both in vertebrates and in invertebrate embryos. Now, so examples of processes regulated by these signaling pathway, including determining the cell types and establishment of cell patterning, which is actually the development of limbs, nervous system, skeleton, lungs, and each these are under the direct control of, so development of limbs, nervous system, skeletal, lungs, hair, teeth, and gonads are all under the control of these pathways actually, majorly hedgehog and Wnt pathway, which are very closely related. Now, the hedgehog gene, which is responsible for this gene is forming a receptor that is called this receptor. Okay, so I will let me scroll this. So see here. So this is your hedgehog pathway inside the cell. So the gene will encode a protein that is shown here in this form. Okay, so this is actually, this is not the blue color. This hedgehog protein is some secreted protein. So this yellow colored protein is called hedgehog protein, which is encoded by this particular gene. Same gene will encode. Okay, so see this first. So first half of this diagram, here there is two proteins. One is called smoothened and one is called patched protein. So this red color is smoothened, whereas this blue color protein is patched protein. Now both are interacting with each other, so they will so you can say they are negative regulators of each other. They will not allow each other to bind with some other molecule and activate. Only when a signal will come, so this secretory protein, which is doing some kind of roles in your early embryonic development of an embryo or some organism. So this secreted protein will come and will bind to this patch protein. After binding, it will undergoing some conformational change, and thus it will be separated from the other protein that is called the main regulator of this pathway, this hedgehog pathway. So this smoothened protein will be freed, and it is now able to interact with some other proteins which are not able to interact with this smoothened protein when it was bind with when it was bound with this patch protein. So now this patch protein is separated, and this is free to interact with certain cytosolic proteins, which were, so this microtubule, this protein is called fused protein, and there is certain other protein costal 2 and this CI-155. So actually the CI-155 is present in Drosophila. In humans, actually the same protein is, the same role is played by some other protein that is called GLI protein. So this is not GLA. Some students may be from GLA, but this is GLI. So this is GLI. So in humans, actually, this GLI protein is present, and this GLI protein is having the same role as in Drosophila is played by the CI-155. So first see this when there is no signal, no hedgehog protein. So this patch is a negative regulator of this smooth protein. Okay, so the smoothened protein receptor, which both are transmembrane receptors, are negatively regulated by patch protein. So not able to interact with the cytosolic protein. So the cytosolic proteins are forming a complex with the microtubules and some other protein, for example, fused costal to CI-155 and okay, so if this type of complex is formed, this will lead to the activation of activation, and this will lead to cleavage and degradation of this particular protein CI-155. So it will leave only one subunit. So this is a dimer. So this is having some molecular weight 155. So if it is degraded, the half will left. So this is CI-75 will left, and one subunit is degraded. Okay, so if the CI-75 will move into the nucleus, so it will repress, it is not going to activate, but further it will repress the certain genes which are under the direct control of this CI-155. So because there is no signal, so this it will generally present in the absence of signal, your cells or the genes which are under the control of CI-155 are repressed by CI-75. But suppose there is a signal, so signal will come and bind to this patched protein. It will free the smoothened protein. Smoothened protein will then interact with certain cytosolic proteins, this costal 2 fused, and most important is this CI-155 in Drosophila, whereas in humans, this particular protein is replaced by this GLI protein. Then this CI-155 in Drosophila or GLI in humans will move inside the nucleus through this nuclear pore complex and will activate the transcription of certain genes. Okay, so remember, if the complete intact protein will go inside the nucleus, it will activate the transcription, whereas if it is degraded or if the half portion or the only one subunit, that is CI-75, will go inside the nucleus, it will repress the certain genes which are under the direct control of this particular protein. Okay, so this is very important and this is very easy too. I hope this is clear. Now we will study whatever is written in this particular text. This is very easy to understand now because at least you know what is happening inside the cell. So in Drosophila there is only one gene, whereas in mammals there are 3 reported genes for this particular hedgehog secretory protein. So it is encoding a secreted proteins that are modified by addition of lipids. So remember, whenever there is an addition of lipids, so it will lead to the formation of protein that is called lipoprotein. So there are 2 types of proteins you have already studied. So antibodies are glycoproteins, certain receptors are also glycoproteins, but it will lead to the formation of lipoprotein because this protein is modified by certain lipids. So this is the lipoprotein. Most important. So the functional receptor of hedgehog consists of two transmembrane receptors, and these are called smoothened and patched, which are interacting with each other, and the hedgehog bind to the patched. Now remember this. So, oh, sorry, this is not working. I do not know why this is not working sometimes. So this secreted protein or lipoprotein hedgehog will bind to a receptor that is called patch. So for remembering this, you actually need to remember one thing. If you heard about pH, okay, so this pH, P for patch signal, P patch receptor, and H for the secreted lipoprotein. So this hedgehog protein will interact with some patch receptor, and it will free the smoothen receptor, which is a negative regulator of this particular second receptor. So this same patch H is also a negative regulator of certain, so this H is a negative regulator of certain uh, receptor that is called smoothen. So it will negatively regulate this particular receptor. So from here you can remember this. Okay, so once this patch is sorry, this is S. No, so this is patch actually. Patch is a negative regulator of this S. So once this secreted lipoprotein, hedgehog will able to bind with this particular receptor that is called patch. It will lead to the smoothened receptor to interact with the cytosolic proteins. Okay, so the binding of hedgehog to the patch allows the smoothen to propagate an intracellular signal. So signal will go inside the cell, leading to the activation of transcription factor that is called CI. CI is a full form. Okay, so this is called Cubitus interruptus in Drosophila, and this is called GLI in mammals. So it is also written here. So this is GLI in mammals. In the absence of this particular lipoprotein, this cubitus interruptus is maintained in a complex that is a protein kinase called fused. Okay, so this fused protein kinase will able to create some phosphorylation over this particular protein and a kinesin related protein. So this fused protein and a kinesin related protein, that is costal 2 actually. So now the question is, what is kinesin and what is the function of kinesin related proteins? For example, this costal 2, which is which anchors the complex to the microtubule. So here, if you notice in this particular diagram, so this costal 2 protein, which is very related to the kinesin family, is able to interact with this microtubule. So in the cytosol, it is forming a complex, and this is a transcription factor, and this is costal 2 is interacting with this cubitus interruptus protein in the Drosophila and GLI in mammals, and also able to interact with the microtubule. Similarly, kinase, because it is a family, it is a costal 2 is related to the family of kinesins. So if you want to know the details of what is this costal 2 and kinesins do inside the cell, you can move to page number 511. So 511. Sorry, actually the page numbers are not same. This is 487. Actually, you need to move to 511. Okay, so here, okay, so here it is showing the kinesin activity. So if you move to the next page, I do not know what. Okay, so this is kinesin proteins actually. So if you look here, this is kinesin. So kinesin is actually the motor protein which will move the cargo, that is vesicles or organelles in the cell, towards the positive pole of this microtubules. So this is microtubule motor proteins. There is two type of motor protein. One is called kinesin, one is called dynein. Now this costal 2 is related to kinesin proteins. So similarly, costal 2 will be able to interact with these microtubules, and it will move this complex towards the positive pole of this microtubule. So it is also written in the text somewhere. If you so this is motor proteins. These are from microtubule motor proteins. So there are two types. One is kinesin. So most kinesins move move towards the positive pole or plus pole, and dynein will move towards the negative or minus pole of the microtubule. So this is the major difference between these kinesins and dyneins. Now we will move again to our page number 635, which is not again the same page because the PDF, this PDF is having certain different pages. Okay. Oh my God, here is my. Fine. Fine. So this is clear now. Okay. So they are related. These costal 2 proteins are related to kinesin protein. So they will move towards the positive pole of these microtubules, and they're called microtubule motor proteins. Now, within this complex, the CI or cubitus interrupters in Drosophila is either completely degraded or cleaved to generate a transcription factor repressor that is called CI75. Okay, after being forming a complex, and in the absence of the signal, it will form CI75 or either completely degraded, so that there is no signaling. Hedgehog signaling promote the interaction of this receptor smoothen with costal 2, and it will lead to the release of a full length CI 155. So you also need to note this. This is called full length CI. So this is having molecular weight 155. So it is able to translocate into the nucleus and will activate the transcription instead of repressing the gene transcription. Okay, it will be an activator. Okay, then there's another family, second family, that is called Vent protein signaling family. So in this particular class of receptors, so first I would like to tell you actually what is happening inside your cell during this Vent signaling. Similarly, in the Vent signaling, as we have studied in Hedgehog signaling, there are two transmembrane proteins. One is called Fritzl and second is called LRP. They are interacting with one secreted protein that is called Vent, WNT, and it will lead to the dimerization. Okay. So in the absence of Vent, they are not able to interact. So all the proteins are forming a complex in your cytosol, where this is a cytosolic protein dishevelled. Axin is forming a complex with APC, and this is called GSK3 beta, which is this is a kinase. So this will phosphorylate your beta catenin so that it will be degraded by ubiquitin mediated proteasomal degradation or by some other means. Once it is degraded, there is no gene transcription. Now there is a second picture which is depicting that if there is a signal bent, it will able to form a dimer, heterodimer of these Fritzl and LRP proteins, and the proteins which are present in the cytosol forming a complex and thus are able to degrade the beta catenin are now able to interact with these, because there is a conformational change after binding of these signal molecules. So these are able to interact with the cytosolic proteins. This will interact with results. So this is FD or DF, whatever you say, and this is AL or LA. So you can write it as, if you want to remember this, so you can write it as FD LA, because this frizzle will interact with dishevelled and LRP will interact with axin protein. And if they are interacting, they are not forming this complex. This complex is not formed, so this particular GSK3 beta kinase will not be activated. So if it is not activated, so it will allow the beta catenin to move inside the nucleus. This beta catenin will interact with certain other transcription factors. This is TCF and LEF. It will activate these transcription factors, and thus the transcription of certain target genes can be achieved. Okay. So let us study and finish this particular signaling. This is called Vent signaling pathway. This is also in certain developing embryos. Okay, so Vent proteins are family of secreted proteins. Again, these are secreted proteins. So let me underline important points. So these are secreted proteins, factors, growth factors that are bind to the receptor that are called LRP. One is called Fritzl family receptors. They're heterodimers. which are related to the smooth family of receptors, which we have seen actually in the Hedgehog pathway. Signaling from LRP and Fritzl lead to the activation of cytosolic proteins that's called disheveled, and inhibition of the complex that is formed of complex Xn, APC, and protein kinase GSK3 beta. Once this complex is inhibited, this inhibition is done. So this is inhibiting the formation of this particular complex so that the beta-catenin will not be degraded inside the cytosol. Within this complex, GSK3 beta phosphorylate the beta-catenin, leading to the ubiquitin-mediated proteasomal degradation. So Wnt signaling result in the, if the Wnt signal will come, it will not allow, it will inhibit the complex formation. It will not allow the beta-catenin to phosphorylate and undergoing ubiquitin-mediated proteasomal degradation, resulting in the increased beta-catenin level. So Wnt signaling is increasing the beta-catenin concentration in the cytosol. This beta-catenin was discussed in chapter 14 also in this book. Okay, so this is a transmembrane protein that link the cadherin to the actin. So what is the function of this beta-catenin actually? So it is help in the interaction of some cadherin proteins to the actin proteins at some specific kind of junctions within your cells that is called AJ, adherence junctions. So importantly, linking the cadherins to the actin is only one role of beta-catenin. So this was only one role of these cadherin proteins, but here this beta-catenin proteins are involved in Wnt signaling, and here these beta-catenin act as a direct regulator of gene expression or gene transcription by forming a complex with members of another family of transcription factors that are called TCF and LEF. So from here, you can note two important functions of the beta-catenin protein. First, they are involved in adherent junctions where they are interacting with the cadherin proteins. They link the cadherin protein with the actin proteins. And here they are interacting with another family of transcription factor that is called TCF-LEF family. And they are able to express the genes inside your nucleus. So these are two very important roles of these beta-catenins. The association of beta-catenin with these receptors, with these transcription factors in the nucleus, TCF, LEF family members, leading to the expression of target genes in the nucleus, okay, in the DNA. Now there is third kind of signaling which is also happening in these embryonic cells. This is called notch pathway, and this is the last signaling actually in which we will discuss in this particular class. Okay, and this will lead to the finish of this particular chapter. So this notch pathway is another highly conserved signaling pathway which control or decide the cell fate inside your animal development or embryonic development. Notch signaling is an example of direct cell-cell interaction. So up to here, the two pathways are very related, in which we have discussed hedgehog and Wnt signaling. Now this notch pathway is different because this is kind of a juxtacrine pathway, a signaling pathway. You have discussed juxtacrine, so in which the pathway in which the interaction between the two type of cells is required. So this notch pathway is actually juxtacrine signaling pathway in which the two cells are interacting with each other during the development of embryo. Okay, so during the development actually. Okay. Notch is a large protein and is a single transmembrane domain that serves as a receptor for signaling by transmembrane protein that is called delta. So notch is having some other protein that is called delta on the surface of adjacent cell. So one cell 1 is having a notch, cell 2 will have another protein that is called delta. Notch and delta will interact with each other. So as you can see from the next diagram. Okay, so what is not happening? So as I move you to the next page, you can see there are two cells. One cell is called, one cell is having delta, and other cell is, this is cell 1, and this is cell 2. And this is the plasma, these are the plasma membranes. One cell is having delta, one cell is having notch. So this notch will create a bond, or specifically able to bind with delta. This is called ND, and this will lead to the activation of one protein, cytosolic protein in cell 2, that is called gamma secretase. This will leave this cytosolic domain of notch so that it will be translocated inside the nucleus, and it will activate some of the transcription factor that is called SU. Okay, so this is also very simple type of signaling. So let me finish this very quickly. We'll move through this. This is very short actually. So during the development and so the notch, one cell will express notch, second cell will express delta. They will interact on the adjacent cell surface. Adjacent cells will interact physically. This is a kind of juxtacrine signaling. Stimulation of notch initiate a novel and direct pathway. This is a direct pathway because two cells are directly interacting with each other. This is transcriptional activation. Okay. So in particular, ligand binding leads to the proteolytic cleavage of notch, and this gamma secretase actually doing this proteolytic cleavage of the cytosolic domain of this notch receptor. Intracellular domain of the notch is then translocated in the nucleus. Notch intracellular domain then interact with another transcription factor that is called SUH in Drosophila, and this is called CSL in mammals. So now you need to note this. This is called CSL in case of mammals and SU in case of Drosophila. SUH. So it is given here. Okay. So it inevitably converts it from a repressor to an activator of target gene. So once the notch intracellular domain will interact with the transcription factor SU and CSL, it will convert these repressors into the activators of these targeted genes, so that the transcription of target genes will be activated. Okay, fine. So notch target genes include the gene encoding the other transcription regulatory protein, which acts to determine the cell fate as a result. So these cell fates will be decided. Okay, so these are major kind of signaling pathways inside our cells. Beside this, there are kind of interactions, and there are signal transduction, which is helping the cell to change the shape of the cell, because they will interact with the cytoskeleton of the cell. And you know, cytoskeleton of the cell is made up of actin, intermediary filaments, and microtubules. Okay, so there are some signaling pathways which are interacting with the cytoskeleton. So one is integrin, as I told you at the start of this class, that we will discuss about integrins. So integrins are of two types. One is alpha, one is beta, and this is given in the. So if you want to study integrins, so actually we need to move. Okay, so there is two type of integrins. Okay, let me find integrin in this. Oh, this is not able to find, but actually these integrins are given somewhere, and at least not in this chapter actually. So let me find integrins. Around page number 580. Okay. So here you will actually these are integrins. Okay, so there are two type of integrins. One is alpha, one is beta. Alpha is having binding site for certain metal ions, and these metal ions are either okay. So somewhere it is given actually. So these integrins are important in cell matrix interactions. So when cell is interacting with the extracellular matrix, so they will require these integrins, and these integrins are also involved in certain cell matrix junctions, and these cell matrix junctions are called focal adhesions or hemidesmosomes. Okay, so one thing was actually I want to show you this integrin structure, and also the catalytic role of these integrins are controlled by certain metal ions, which are either calcium ions, magnesium ions, or manganese ions. Okay, so okay, actually it was given somewhere, but I forgot where it was written actually. So let me scroll this. So actually these are interacting, so the cell is interacting with the extracellular matrix with the help of these integrins. So you can see here, so whenever there is focal adhesion formation, so there is a regular interaction between these integrins, and there is a cell, there is an extracellular matrix. Also, you can see it from this particular diagram that these cells are forming either focal adhesions or hemidesmosome, in which integrins are having some active roles. For example, integrin alpha 6 and beta 4. So in hemidesmosome, it is shown here. There are certain other regulatory proteins also. For example, binding protein 180, binding protein 230. So okay, so they are also helping. So the major regulatory protein in this case, hemidesmosome, is binding protein 230. There's certain other protein plectins, laminins are also there, so that the proper interaction between the cell and cell matrix is happening. In focal adhesions, there are proteins called talin proteins, vinculin proteins, and alpha-actinin proteins which are interacting directly with the actin filaments. So these are actin filaments, and they are interacting with the alpha-actinin and other type of proteins, talin and vinculin, so that they are controlling overall the cell shape and movement of the cells in the tissues. Okay, now we are coming back to our class. Okay, so here this actually we have discussed. So here also it is shown these are integrins, and they are interacting with so many type of molecules. Okay, so now so these integrins are major receptors responsible for attachment of the cells with the extracellular matrix, as we have seen, at two types of cell matrix junctions. These are called focal adhesions and hemidesmosomes. I showed you. The integrins also interact with the components of the cytoskeleton to provide a stable linkage between the extracellular matrix and the adherent cell. So in addition to this structural role, integrins serve as a receptor that activate the intracellular signaling pathways. So actually, this was the first role. Actually, it is also having certain other roles. So as you've seen this, so first thing, it is helping in cell matrix junctions. The two type of major junctions are focal adhesions and hemidesmosomes. Whereas it is also having some structural roles. It is involved in intracellular signal transduction pathways, thereby controlling the cell movement. So that cell movements and cell shapes are other aspects of cell behavior. Example, proliferation and survival are also under the control of these integrins. Okay, so like members of cytokine receptor superfamily, as we have discussed earlier, the integrins have short cytoplasmic tail, but these do not have any intrinsic enzymatic activity. Okay, these do not have these activities. However, protein tyrosine phosphorylation is an early response to interaction of the integrins with the extracellular matrix component. This is suggesting that integrins are linked to the non-receptor tyrosine kinases in a similar way जैसे कि हमारे receptor tyrosine kinase interact करते थे और non-receptor tyrosine kinases. One mode of signaling downstream to the integrin involves this protein that is called kinase, that is focal adhesion kinase. Okay, so this is focal adhesion kinase. Beside this focal adhesion kinase, as its name implies, focal adhesion kinase is localized to the focal adhesions and it is rapidly become tyrosine phosphorylated following binding of integrin to the extracellular matrix component. Extracellular matrix component, for example, fibronectin. Like other protein tyrosine kinases, the activation of this focal adhesion kinases involve autophosphorylation because these are tyrosine phosphorylation is doing induced by clustering of integrin bound to the extracellular matrix as well as their interaction with the actins. So as I showed you in the case of focal adhesions and hemidesmosomes, they're interacting with actin. Okay, autophosphorylation of this kinase create docking site because they're creating phosphotyrosine docking sites for signaling molecules which are having this domain because the SS2 containing protein can directly interact with these phosphotyrosine residues, including members of SRC family. So these SRC family protein can interact, these are having SS2 domains and these can interact with phosphotyrosine residues that phosphorylate additional sites in these focal adhesion kinases. So they will also phosphorylate the additional sites in these kinases itself. As discussed earlier, for the growth factor receptor tyrosine phosphorylation of these kinases create binding site for SS2 domains of the other downstream proteins. So many proteins can interact with these SS2 domain containing protein, including phospholipase C gamma, PI3 kinase, and GRB2. This is adapter protein we have discussed in yesterday's class. All these are having these SS2 domains, therefore they are able to interact with phosphotyrosine and this focal adhesion kinases. So now I will move you to the next page where it is written or shown clearly. Okay, so see here, these are focal adhesion kinases and these are integrins. So these are intracellular kinases. They are interacting with the integrins and this is extracellular matrix. So they will phosphorylate each other on tyrosine residues. Then these phosphotyrosine residues will create a binding site for the downstream molecules where they will also phosphorylate the additional sites of these kinases itself. For example, SRC family members. Beside this SRC family, other proteins, for example, PI3 kinase or the GRB2, which is the adapter protein, which is having SS2 domain, two SS3 domains. So it will interact with some SOS protein. This SOS protein is a guanine nucleotide exchange factor. So it will activate the RAS protein. RAS protein will activate some MAP kinase pathway through RAF, MAC, and ERK. And finally, this MAP kinase pathway will activate certain genes in the cytosol, in the nucleus. There is PLC gamma is also activated as you shown in case of receptor tyrosine kinases. So this will activate the calcium signaling pathway or diacylglycerol, or this is called protein kinase C pathway. Okay, so these integrins, which are having two type of functions, they will interact with the extracellular matrix, are able to transmit the signal inside the cell, and they will act via various mechanisms, either by calcium signaling or protein kinase C signaling, or via this is called protein kinase B signaling, or protein this PI3 kinase pathway, or via the MAP kinase pathway. So they can go through any pathway, and thus they can interact or they can transmit their signals. They will allow the certain genes to express inside the cells. Now the second type of interaction or regulation is of the actin cytoskeleton. So these actin are the major part of our cell cytoskeleton, and these actin proteins are actively involved in the cell movements actually. So cell will move. So motility of the cell is due to these actin filaments. So signaling from the integrin as well as from the growth factor receptors also plays a central role in the control of cell movements. So whenever there is the cell want to move, so there is signaling called the signaling is through actin cytoskeleton, and the cell movement by regulating the dynamic behavior of actin cytoskeleton. Okay, so cellular responses to growth factors as well as cell adhesion receptor frequently including the changes in cell motility, because the cell movements are under the control of these actin cytoskeleton. As you can see from here, there are three family of Rho family kinases. You also studied this from your Pathfinder actually. So here, actually they were talking about three type of kinases. So if you do not remember, I will lead you to this particular discussion. So this is called Rho family, and this is given in the MAP kinase pathway. So beside RAS proteins in MAP kinase, the other class of RASPER family GTPases are three families in which the Rho itself, there is a RAC and there is CDC42. Now in today's class, we will discuss what are the functions of these three families, members of the Rho family, or which are coming under the direct in this MAP kinase pathway, which are very related to this RAS protein, because this RAS protein is a GTP binding protein. So now they're having different functions. The CDC42 is involved in filopodia formation. RAC is involved in lamellipodia formation, which are the cell surface appendages actually, filopodia and lamellipodia. Then this Rho, which is also a GTPase or GTP binding protein, is also involved in formation of focal adhesion and stress fibers. So these focal adhesions are the points where the cell are interacting with the extracellular matrix, where these stress fibers can be seen inside the cell cytosol. So these are under the control of Rho family of proteins. Okay, so it is given here now. Members of Rho subfamily of small. So I will let you to discuss. Actually, this is okay. So cell motility or the cell movements are under the control of these CDC42, RAC, and Rho. Either they're moving. This is filopodia, lamellipodia, or Rho is actually forming the stress fibers. So as we have seen in this particular diagram, these play central role in regulating an organization of the actin cytoskeleton. So the major role of these proteins, these three proteins, Rho, RAC, and CDC42, plays a central role in regulating the organization of actin cytoskeleton and thus control of variety of cell processes, including the cell motility. Because here, this actin filament or actin cytoskeleton is majorly involved in this cell movements and the cell adhesion also, because the focal points are the points where the cell is, with the help of these actin filaments, the cell is interacting with the cell matrix. The role of Rho family members of the cytoskeleton remodeling was, so they are involved in remodeling of the cytoskeleton or the actin filaments, demonstrated by experiments showing the microinjection of fibroblasts. So this is shown that certain experiments were done where the microinjection was given to the fibroblast cells, and it was showing that there is a Rho protein which induce the cytoskeletal alterations, the change in cell shape or movement or the change in position of the cells, including the production of cell surface appendages or protrusions. Protrusions are called filopodia and lamellipodia, whereas the formation of focal adhesions, stress fibers. Microinjection of cell with the specific mutants of different Rho family members shows that if we give the specific mutants in the microinjection, that so that the specific structures will only form. Okay, so either filopodia, lamellipodia. So this CDC42 was found to the involved in the formation of filopodia. RAC is mediating the lamellipodia, and the Rho is responsible for the formation of stress fiber inside the cell. Further studies have demonstrated that the activities of Rho family members are not restricted to the fibroblast only. So this is the experiment, initial experiments were done in fibroblast cells, but later it was found that it was present in all type of eukaryotic cells. Okay, so they are having some kind of global expression. Rho family members are activated by integrin signaling as well as by the growth factor receptors. So these integrins are signaling, as we have seen. These integrin signaling can be via MAP kinase pathway, via calcium signaling, or this is called IP3-DAG pathway, or via some receptor tyrosine kinases. So these integrin signaling as well as by growth factor receptors. So Rho family members are very similar to the RAS family members, and they can do signaling either via integrins or via the growth factor receptors. A large number of proteins that serve as mediate the cytoskeletal changes induced by Rho, RAC, and CD42. For example, a key target of Rho is regulating cytoskeletal alteration protein CD3 and kinases called Rho kinases. So now you can see how the regulation is being done. So there is a Rho protein. This Rho is controlling the Rho kinase. Rho kinase is inhibiting the myosin light chain phosphatase. So it will not allow the phosphatase to remove the phosphate group from the myosin light chain. Also, it is phosphorylating the inactive myosin to convert it into the active myosins, because it is phosphorylating the regulatory light chains of myosin. If it is phosphorylated, so myosin will be activated. So on the one hand, it is activating the kinase so that the light chains can be phosphorylated and activated. On the other hand, it is inhibiting phosphatase so that it cannot remove these phosphate moieties from the myosin light chains, which are having regulatory function. So this is under the direct control of these Rho family kinases, which are very central role in regulating these type of interactions in myosin 2 chains, light chains of myosin 2. So because the myosin 2 light chains are having some regulatory roles. Oh, sorry. Okay. So actually this is finished. In tomorrow's class, we will discuss certain type of networks and crosstalks which is being happening between different type of receptors. So these are called feedback mechanism, feedback loops or crosstalks, so that they can form certain type of networks. So these networks are very common in cell signaling, in which either it is a negative feedback mechanism, positive feedback feed for forward relay. There is stimulatory crosstalk or inhibitory crosstalk between different type of molecules and signaling pathway. And different signaling pathway, if come together, they will lead to the formation of signaling networks. So this kind of networks are present inside your cells. So suppose if there is all the signaling happening at the same time in a single cell, so if in a single neuron there are reported 545 signaling pathways, in a single neuron you can report 545 signaling elements in a single mammalian neuron. So it will lead to the formation of this type of junctions where all the signaling pathways are crossing each other. And these signaling pathways may be of three types, where these green arrows, if you are able to see some of the green arrows, so green arrows are stimulatory. They will stimulate the signal transduction. Red arrows are inhibitory and blue arrows are neutral links between different type of signaling pathways. So this is actually happening within our cells. So you can think of how complex cell signaling is always happening inside our cell. And if everything is okay, and there is no problem. If something happening, okay, so something is not regulated inside the cell, so it will lead to the disasters, for example, cancer or tumor formations. So you need to keep in mind that not only a single pathway, we have studied in our class, for example, MAP kinase or GPCR pathway or some other calcium IP3 DAG pathway is not happening inside the cell. All the pathways are happening in, they are crossing each other, they are inhibiting each other, they are stimulating each other, they are talking with each other. So this is all happening in our complex cells. So the complexity of signaling in a single cell is very, very high, and there is a requirement of proper regulation so that everything can be maintained and regulated properly. So with this, I'm closing today's class. So hope this is helpful, whatever we have discussed in today's class. For a quick summary, whatever we have discussed. So we have discussed many things in this particular class, started from JAK-STAT pathway of cell signaling. We have discussed JAK-STAT. We have discussed TGF-beta-SMAD pathway, transforming growth factor beta-SMAD pathway. So transforming growth factor beta-SMAD pathway is made up of heterodimer, in which second is activating phosphorylating 1, and this is a serine kinase, and the SMAD protein is a transcription factor going inside the nucleus. Then we have discussed about NFkB signaling, which is a kind of inhibitory protein. If this is phosphorylated by the signal will come, and this kinase will phosphorylate this. This is having specific kinase, so it will specifically phosphorylate the I-kappa B. This inhibitory protein kappa B will be phosphorylated and undergoing the UB cutinated, and this is called ubiquitin mediated proteasomal degradation of this particular I kappa B. It will free the NF-kappa B, so that the NF-kappa B form a heterodimer and will go inside the nucleus. Translocation will happen, and it will thus control the certain gene transcription inside your nucleus. Then we have discussed three pathways of cell signaling which are happening in early embryos, and they will decide the cell fates. For example, developing cells. Hedgehog went and notch pathway in hedgehog signaling. This is a lipoprotein. If it will bind to the patch, they have discussed pH here. So this patch is bind with hedgehog. So it will free the smooth hunt. Smooth hunt will interact with certain cytosolic proteins fused, and costal 2 will interact with the CI155. The CI155 is having another protein in our humans. This protein is called GLI in humans, and in humans, this GLI will go inside the nucleus and activate the transcription of certain genes. Then, okay, let me finish this. Then we have discussed vent signaling. Vent signaling is having two type of receptors over cell surface. They are forming heterodimers. Vent signal will come, which is a secretory protein again, and it will allow the dishevel to interact with the frizzle. So this FD will allow the axon protein also interact with the LRP, and this will allow the beta catenin concentration will increase in cytosol. It will not be degraded as in the absence of signal. So it will go inside the nucleus, it will interact some other transcription factor TCF and LEF, and will activate the transcription of certain target genes. Then we have discussed the notch signaling. Notch signaling is a kind of juxtacrine signaling in which the two cells are required. One cell is having notch, one cell is having delta. Delta will stimulate this notch. There is a conformational chain, then it will activate some secretase enzyme, gamma secretase, so that it will kill the C-terminal domain of notch, so that the C-terminal domain will be transported into the nucleus, and it will activate some of the transcription factors, for example, SUH in Drosophila and CSL in mammals we have discussed. Okay, SUH in Drosophila and CSL in mammals is activated, and the transcription of certain target genes are thus. Then we have discussed about integrins. Integrins are having two type of roles. They are either interacting with the extracellular matrix, or they are involved in the cell signaling by various means, not only by the SRC proteins. They are also involved PI3 kinase mediated signaling, or the protein kinase B AKT signaling. They are also doing signaling by PLC gamma, and this PLC gamma is going to activate some of the phospholipase C. Gamma will do signaling by calcium mediated pathway or IP3 DAG pathway. Then there is a signaling by these adductor molecules. These are called GRB2. So this GRB2 is adductor which will activate some of the guanine nucleotide exchange factor SOS protein. This SOS protein will lead to the activation of MAP kinase pathway. Then we have started the regulation of actin cytoskeleton, and cell movements are under control of actin. And these actin filaments are also involving these Rho, RAC, and CDC42, which are respectively involved in the formation of lopodia, lamellopodia, and focal adhesion and stress fiber inside your cells. This Rho is under the control of Rho kinase. So, and this Rho kinase is responsible for phosphorylating the regulatory subunit of myosin 2, which is okay. So that's all for today. We are running out of time, and I do not know how many members are there in class. Only 4 members are there: Arti, Amu, Nidhi, and Priyanshi. No problem. So we have discussed so many things in today's class. If you face any query or you have doubt, you may ask. I'm unmuting you all for some time so that you can ask your queries, and then we will close today's session. Okay. So